Welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society, and I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. The views and opinions expressed by our guests are their own and do not necessarily reflect the views of the host, the American Phytopathological Society, or any affiliated entity. This is the 5th episode of 2026, and today I'm excited to welcome Dr. Melanie Medina López, postdoctoral researcher in the Department of Plant Pathology and Environmental Microbiology at Penn State in University Park, Pennsylvania. Dr. Medina López holds a BS in Industrial Microbiology from the University of Puerto Rico, Mayagüez, and an MS and PhD in Plant Pathology from The Ohio State University. Dr. Medina López joined the Department of Plant Pathology and Environmental Microbiology at Penn State in 2025, where she's currently a postdoctoral researcher. Her work is currently focused on comparative genomics of plant pathogenic bacteria and plasmidomics. She previously served as a postdoc in the Bull and Kovac labs prior to her current position. There, she worked on microbial predators and the roles in co-mitigation of plant and human pathogens in the soil. Dr. Medina López's graduate research focused largely on soybean cyst nematode and their associated phytobiomes, having published this work in Phytofrontiers, Phytobiomes, and Applied in Environmental Microbiology. Dr. Medina López previously taught advanced phytobacteriology lab. She's given guest lectures for general plant pathology and introduction to microbiome analysis, and served as workshop instructor or helper for workshops on metagenomic analysis, introduction to R, and network analysis. She's an active member of APS, ASM, AAAS, and the Soil Ecology Society, as well as the Society for the Advancement of Chicanos, Hispanics, and Native Americans in Science. Welcome, Dr. Medina López.
Thank you for having me. This was a pleasant surprise to be able to join here.
Yeah, absolutely. It's always exciting to look over someone's CV or resume for someone that you don't know, because you're like getting to know all about them and their academic journey and like their systems and pathogens that they work on. I see that you, in addition to holding both a master's and a PhD in plant pathology, which is not surprising given that you're a plant pathologist, you also have a BS in industrial microbiology from the University of Puerto Rico, Mayagüez campus. Did I say that correct?
Yes, that's correct.
Are you from Puerto Rico?
Yes. Nobody can see this, but I have a little flag, mandatory flag if you're Puerto Rican. We always have one like nearby in case of emergency.
Where on the island did you grow up? And I know, of course, that your institution is on the western coast. It's one of 11 campuses of the University of Puerto Rico. Yours is second only to the campus that's in San Juan. So where did you grow up in proximity to that?
So, fun fact, the University of Puerto Rico Mayagüez is the only land grant university in Puerto Rico. In the beginning, it was very focused on agriculture and then expanded into other things. But I grew up in Isabela, which is Isabela Quebradillas. They're side by side. There's two small towns in the northern coast of Puerto Rico. So I grew up near the beach.
Okay.
And it's about an hour or an hour and a half away from campus. So I didn't commute to campus. I started there and stayed, visited my parents on the weekends. It's a very fun campus if you're an undergrad. And the town, it's your typical college town. So whenever class is in session, it's full of life. And then once it's break, there's no one there.
Yeah.
But yeah, I did my undergrad there in industrial microbiology. I didn't start in industrial microbiology. I started in biology, actually, because I wanted to be a doctor.
I sense that your involvement in the medical association for like all your years as an undergrad, I saw that, made me think that you were interested in pre-med or that, and then at some point you switched. So I was going to ask you about that, if you wanted to expand on that. But industrial microbiology, is that a larger major there, or is it just one of many biology-focused or biology-centric majors at this university?
So the Department of Biology in the university, at least in Mayagüez, it's one of the biggest departments. Well, it's the most students. but it has the bachelor's in biology and in microbiology. And I think at the time there was also an undergraduate degree in pre-medical sciences, something like that. But the biology major, I think it's the bigger one, but it's mostly composed of people that want to go to med school. So there was a lot of opportunities to be involved with medically related associations, but also a lot of clinical associated micro classes. So you had your medical mycology, you had your, sometimes they give epidemiology, sometimes they give embryology. So things that would help you if you were on track to be a doctor
So
down the line.
let's rewind a little bit. So obviously you grew up north of campus by an hour, you said.
About an hour away,
So
yeah.
what was life like in the town where you grew up? And I know you were thinking pre-med and that, but was it rural? Was it a bigger agricultural area? Obviously the land grants there, which means that there's got to be like experimental farms and things like that nearby. Break it down for us. And that part of Puerto Rico is, what are the main crops and what did you see growing up?
Honestly, Puerto Rico doesn't produce a lot of the food that it consumes.
Sure.
One of the primary crops are going to be your plantains and your coffee.
Yeah.
Okay, those are the things that are primarily grown there. And interestingly, unless you grew up in more towards the center of the island, a lot of people are removed from like cropping
And food
systems.
production.
Yes.
But I've been to some of the coffee plantations there. I was last in Puerto Rico in 2018, and I guess this would be like a year after Hurricane Maria hit. And obviously, there was a lot of. I went to El Yunque and National Forest while I was there too. But I saw some of the coffee production firsthand, and of course, as a coffee drinker myself, I appreciated that. But something I was thinking about as I was looking where you went to school and just having family there is just how much your life has been impacted by hurricanes. I'm
Oh,
just
yeah.
I'm just thinking about the timing. You weren't yet in the United States studying when Maria hit, and I couldn't imagine the devastation that had and the impact it had on you as a younger individual.
I was still in undergrad when Maria happened, and I had to make a decision. Do I stay on campus in my apartment because I was living there for the hurricane, or I go back home to wait for it to pass there? And I decided to stay on campus
Yeah.
because all of my things were there, and I'm like, if something happens, I want to be there. And I was not able to go back home for about a week, 2 weeks, because the main road to go back home
Yeah.
was like completely
Covered in
inaccessible.
trees, and washed
It
out and all
was,
that.
yeah, it was just dirt covering the whole thing. So we had to wait about a week or a week and a half for one of the main roads to be cleared for me to be able to go back home. And we didn't have access to cell phones or anything. So we were really struggling to figure out, okay, is it only inaccessible in this part? Am I going to be able to drive the whole hour away
Yeah.
or not? And then for classes, that was weird because there was no power.
Yeah.
And the university had no power for a while. So it was prioritized to give energy and water to those areas, but at the same time, the students living surrounding the university, most of us didn't get power or water service for months. So
Yeah.
you were having to go to school, you had to go to college before you had all of your basic
Needs being met.
needs. Exactly.
Yeah. I think it's important to talk about it because it's like, as we think about food security and we think about plant health, you can't not think about abiotic agents. You can't help think about extreme weather, resilient landscapes. Even if they're resilient and you're building resilient soils and resilient landscapes, it's really hard to account for these really adverse weather events, hurricanes, tropical storms. And as someone like you that grew up not only Maria, but storms before that, and then the 90s and the after that, constantly just trying to live your life and under the threat of these potentially landscape devastating storms. And we're facing those kinds of problems in our agricultural areas. So you have a much sharper and well-rounded perspective on these things.
And I think you learn a lot about why evolution happens in the way that it does, right? Because for a lot of the fruit trees and other crops that we are used to, because sure, we're not really tied to agriculture, but I think everyone's familiar with having that huge fruit tree in their backyard.
Sure.
The grandparents put there years and years ago. And you see how they're bare after the storm, but they recover fairly quickly. And next year you have fruits again.
Yeah.
And most of these are like introduced, they're not native, but still you see how just the climate of the places where they're originally from shaped their resilience to be able to sustain these extreme events.
Yeah. And also can't help but think about the inadvertent movement of vectors and vector-borne diseases on these strong winds and things like citrus greening and some of these other things that really impact tropical fruits and even citrus production in places like Florida, and has pushed citrus production into other parts of the US like Arizona and Texas and California because the landscape can no longer support a sustainable fruit tree agriculture or citrus production in those areas. But it comes back to this understanding of how these extreme weather patterns are shaping our where we conduct agriculture and where we can continue to think about a future in agriculture.
Yeah. And I think it's also hard to communicate these things to people in ways that are accessible, right? For example, you mentioned citrus screening, and for a long time, my grandpa, he has a lot of fruit trees in his backyard. And I'd go to his house before I knew anything about plant pathology, and he would say, "Man, my orange tree, my lime tree, they're dying, and I don't understand why. I haven't changed anything." And at that point, I'm like, "Huh, that's odd. I don't know anything about this."
Yeah.
And then when I started studying plant pathology, I'm like, "Wait a minute. I know what this is now. I wish
Yeah.
I had known back then so that maybe some of those trees would be alive today. We had been able to manage in time." I think people would get a lot of use out of just knowing more about plants and
Yeah.
plant pathology.
I want to talk about some of your early experiences before plant pathology. And I saw that at UPRM, you worked on processing mesophotic reef samples from the
Yes.
Caribbean. Can you talk about what that work was like and what you were researching exactly?
I had a great ecology professor. She's a marine biologist, and she was studying some of the smaller animals that live in the sand, basically, to understand whether the species that were there were any different from species you find in like full light reefs. So my job, which prepared me for my later work in nematology, was taking sand samples, right?
Yeah.
And putting them in a dissecting microscope and just searching for tiny animals, picking them up with tweezers and separating them for a graduate student to have the lovely job of identifying the taxonomy, like
Mhm.
where they belonged, and whether any of them could be a new species.
Was that like both like classic morphological identification, but also like sequence based or?
Yes.
Okay.
So they did both. I worked on the front side of it. So the idea is that once the undergraduate did the grunt work of separating the little animals, they would go to an experimental station that did the sequencing and understanding how all of that was done. But by that point, I was very close to graduation. So I was like, I really need to focus on what I'm actually going to do after this.
Yeah.
So it's not going to be in my best interest to just continue putting more time into this research, because at that time I was interested in grad school. So I was looking for opportunities that were a bit more relevant to what I wanted to do in the future.
Speaking of kind of grad school, we'll get there, but upon graduation in 2018, you came to the Ohio State University Worcester campus for their summer research opportunity program.
Yeah.
Um, Now, according to what I read, you were working on some assessing nano vaccines for influenza there, which is again another pivot, but again, like just putting yourself out there and getting an opportunity to come to Ohio and do that. You want to talk a little bit about that and what you got out of it?
Honestly, I tell people I'm just a nerd. I just like science. I was exploring, I was looking for what is it that I want to do with my life, because after I decided I didn't want to be a doctor, which is another tangent and interesting story about how I found out I did not want to be a human doctor or a
Well,
doctor for humans.
you're welcome to share it if it's relevant, but otherwise you can continue on.
So I was in industrial microbiology at this point, which is a lot of figuring out whether you want to go into development of drugs or development of strains for production of things like food or different chemistries. But one of them, it's like development of vaccines. So I tried that at Ohio State. The idea was that so when swine gets the flu, they just get you get reduced weight, which is bad because you want big, juicy swine
Yeah.
to eat. So they were looking for a way to elicit a good immune response to be able to combat influenza in the system. So we were testing vaccines that were delivered nasally. And at that point, my job was basically to run the tests that were going to determine how strong that response was. So it was ELISA, which plant pathologists also use. And that was pretty much what I did during that summer. It was interesting. I learned a lot and it helped me get my first job out of undergrad. But I also learned that I did not want to work with animals either.
Okay. But you had to have that experience to figure that out, right?
Yes.
Part of our academic journey is to go after something, realize it's not for us, and then change directions, right?
And that's one
So,
thing I love about working with undergrads and helping them explore what is it that they like. I'm always telling them, if you have absolutely no idea what you want to do, just say yes to as many things as you can in terms of opportunities that you come across, because you never know what's going to be that point where you say, oh man, I didn't even know this existed, but I love it.
yeah, absolutely. And it's humorous sometimes. I love enthusiasm of really early career undergrads and the first research experience, and they're like, I'm going to do this. And I'm like, that's great if pursue that, but you'll realize that as you experience and add more things to your resume, that your eyes will be opened up to this thing you didn't even know existed. So less than 2 years later, upon doing this summer research opportunity program, you start as a grad student at the Ohio State University Worcester. And I can't help but think the two things are linked. You stay in Columbus and you work, is it at Battelle Memorial Institute? Did I say that right?
Yep.
That's in Columbus. What did you do there? And like, how did you go from this summer research opportunity program to getting into and becoming a grad research fellow?
So during the time I was in that summer program, I made a lot of friends. If people are not familiar probably with the Worcester campus as much as they may be familiar with the Columbus campus at Ohio State, but Ohio State has different campuses, right? Worcester is one of them. And Worcester is primarily research. So there's ATI that has their technical side and more shorter programs. But then The rest of the campus is research, it's grad students. So when I did my summer internship there, I met a lot of grad students and they became my friends. So when I moved to Ohio to do vaccine development research, I kept those friendships going. During my time at Battelle, it was right at the time of COVID, which meant that I just started working on the first assay developments to really understand how we were going to test the efficacy of the vaccines.
So you were still doing vaccine development there
Yes.
at the institute.
Yes. My research during the summer really helped me to get that job because I already had a little bit of experience working in that. But the cool thing about being in Ohio is that I could connect with the people I had met during the summer. So I just visited them. I lived in Columbus, but I went out to Worcester to party with the friends I had made during the summer.
Sure.
And at that time, one of the people that was there, we were talking, and she was talking about her research, and I was like, oh, that sounds so great. I would love to do that. And she was like, my advisor is going to be looking for a grad student really soon. And since you did this summer program, you're eligible for a fellowship and maybe you get it. Who knows?
Yeah.
That would help a lot. So I connect with the advisor, Soledad Benitez Ponce. She's been on the podcast before. And we talk and I'm like, oh, this research is so cool. She was working on microbial communities in the soil and how different cropping systems change the microbial community and how we can manage pathogens using microbes and all of that. And I'm like, oh, sounds amazing. I would love to work on that. She's, let's see, you apply and we'll see what happens. From all of this, it's like, this is why I think just exploring a lot helps. You never know which friend, which connection is the person that's going to lead to your next opportunity.
Yeah.
And also, if people invite you to a party, just say yes. Just go. You may end up finding your advisor through someone that went through that party.
Yeah. And having interviewed a number of people now and being in my third year here as host, I hear a similar story where people, whether we're talking about people that are just moving across the country or people that are moving from South America or wherever, they take a chance, they make those connections, and then, of course, they leverage those connections for their grad school experience or their postdoc. And that's what those experiences are for. Yeah, they're meant to give you some experience, but really, like so many things, it's about the network that you develop. You start as a grad research fellow, and this would be in fall of 2020, right?
Interesting time to start grad school.
Yeah. And you end up getting both a master's and a PhD in plant pathology. Were your thesis and your dissertation on the same topic? Were they both on the soybean cyst nematode?
Yes. Ohio State does this interesting thing where you start the program. I started as a master's student. My idea was to do a master's and try my chances at the job market, see what happens.
Sure.
But at the time when I think it was around the second year mark when I was pretty close to finishing what would have been my master's, my advisor said, listen, I think you'd be great as a PhD student. If you're interested in staying on, you're welcome to do. If not, you can still change into the PhD program. And if you pass your candidacy, Ohio State can give you the master's degree. And then you can decide whether you continue to your PhD and include all of the research that you did during your master's within your PhD, or if you master out and it's like you have the choice to do that. So at that point, I'm like, I would like to do a PhD. So also the world was at an interesting point because it was like I started grad school right in the middle of COVID, and 2 years in, it was still a bit difficult
Yeah.
to make a decision as to whether I wanted to just leave with a master's or continue a PhD. And I was having fun.
Yeah.
I was very interested in what I was doing and was very passionate. So I was like, I'll just keep going. So I passed my candidacy. I get my master's, but I continue the work. And during that time, I worked with this soybean cyst nematode, which, if you're a soybean farmer, you probably know what that is. But if you're not, it's basically a nematode that goes into the roots of soybean plants, and it causes yield losses primarily. And it's like a silent killer. You don't notice it's there until it's a bit too late.
By the time symptoms, you're already in trouble.
Exactly. And it takes a long around 6 year process to, if you have very high levels, to get back down to a point where you're like, okay, I'm not losing more yield to this nematode. So we started just doing a survey. It had been a while since Ohio had been surveyed for the nematode, or that all the data that we had available was put together into a survey. And during that time, the project was already ongoing when I joined. So I got handed a bunch of samples. Right about I got there, there was nobody in the lab because only one person could be in the lab at a time. And my advisor was like, I'm so sorry, but here you go. Try to figure it out.
In a sense, that's a good thing because at least you'll have like the first chapter. You're getting handed samples that are already ready. All you have to do is process them and analyze them and do all the computational stuff that you did. But yeah, we've had a couple of guests over the years that have worked on nematodes. But just to remind our listeners, we're talking about a group of microscopic, free-living, and parasitic roundworms. This includes things that cause heartworm in dogs, trichinosis, pinworms that humans get, but what we're talking specifically are about plant pathogenic nematodes, which include the soybean cyst nematode and another nematode that can be mistaken for it, but we deal with a lot of crops, is the root knot nematode. But back to your soybean cyst nematode work, obviously soybean production is really important in the United States. I don't know where Ohio ranks in terms of production, but it's probably up there. Tell us a little bit about this. So you get these samples, but where does the project go from there?
The idea of the project was to see if we could find any fungi that could potentially help us control or manage the nematode. So basically a biocontrol agent. That was the initial goal of the research. We sampled with a lot of help from a lot of the extension agents within the Ohio State's system of extension. They helped us identify farmers that would be willing to send us their soil. And with that soil, we looked at how much nematode was in the soil and also which fungi were in the soil. And we initially thought we will focus on the samples that have low nematodes to see if the fungi that are in there could have any potential to be reducing the abundance of nematodes in that field. But what we found was interesting because we found the opposite effect. We
And
found,
explain this, and this is your phytobiomes paper
yes,
on the fungal communities associated with the soybean cyst nematode. I read the paper; it was really interesting. But please tell our listeners what you found.
what we found is that the more nematode you have in your soil sample, or at least the samples that we received, the higher the abundance of fungi that we'd known in the past have negative effect on nematodes. They eat nematodes or they destroy their eggs, those sort of things. And in the beginning, it's very counterintuitive. And we were thinking, like, that doesn't make any sense. If you have more of these fungi that, in theory, should be eating these nematodes, why would you have more? Shouldn't they be less? But we started thinking a bit more broadly about the ecology of these fungi and their relationship to the nematode. And the concept of suppressive soils really helped us in framing the way we thought about our results. So, for those that don't know, suppressive soils are pretty much what the name suggests. They're soils that either because of biological reasons like a microbe or things that have to do with the actual physical properties of the soil have the effect of reducing the damage a pathogen can do in a plant or reducing the abundance of a pathogen in that soil. And what we know about the way that the biological type of driven soil suppression is that you generally need a high pressure of the pathogen. So your pathogen has to be abundant and causing significant problems for a long time before you'll see the buildup of this suppressiveness. Then you'll see a sudden drop in the damage of the pathogen to the crop. So we're thinking, okay, wait a minute. If we think about suppressive soils and we look at our results, things start to make sense. So when you have higher levels of the nematode, you're creating this pressure that starts to select for the things that eat the nematodes. If the food source is there, odds are that the things that eat it
Yeah.
are going to increase in abundance.
Because with the disease triangle, it's not just about having a susceptible host, it's having an abundant amount of the susceptible host so that a biocontrol agent such as a fungus can actually gain enough momentum to cause disease, right?
Exactly. So in theory, if we just let the nematode do its thing for 30 years, we may see a suppressive soil down the line. But while we think it's happening, we're getting a snapshot of the point where the nematode is in high enough abundance for these antagonistic fungi to also be in high abundance, but not to the inflection point where they consume enough of the nematode for the population to drop off.
And the other thing is, when I looked at your Venn diagram, I saw that fusarium, the one that's associated with the disease of soybeans, was there. And of course, that's going to be a problem because even if you're controlling the plant pathogenic nematode, you're still dealing with a fungal plant pathogen that itself can cause pretty devastating disease. So it's like the additive properties of these two pathogens where you're trying to control one, but you still have to deal with the other. It's complicated.
Yeah. And from that project, we started thinking, okay, then what's like the next logical step? We've seen how this distribution, this association happens across the state of Ohio. So we've started thinking about, okay, can we detect this also at the field level? So the next project that was a continuation of these initial findings was trying to see if the effect also tracked within a field. So the way that nematodes are distributed within a field, and really a lot of plant pathogens are distributed this way, but they tend to have hotspots in different locations on the field. So our hypothesis based on what we had seen is that within these hotspots, we would find higher abundance of also the fungi that are associated to or that are antagonistic to the nematode. So we enlisted the help of a very lovely farmer that let us just go into his fields for 2 years and do very extensive sampling of his fields. This is some research that we are working on publishing, hopefully soon, but who knows, where we did a gridded experiment. So he had 4 different fields in a corn soybean rotation, and we split his fields into 1 acre grids. And we sampled those fields in all of those grids. And within that acre, we would collect rhizosphere and bulk soil samples and do basically the exact same process of counting the nematodes and identifying the fungi in those hotspots. And one additional thing we did is that we also started building a collection of fungi from the hotspots. For this project, we had the help of an amazing undergraduate student. Her name was Ananaya Cohen. And she basically helped me build the fungal collection.
Yeah.
We developed together a high throughput isolation process. For those of you that worked with fungi, you may be familiar with the difficulties of trying to isolate a lot of fungi from a very hyper diverse environment like soil, where
where trichoderma and penicillium just grow on everything.
Yeah, and fusarium. Like our soil's filled with fusarium. And if you have your plates and something sporulated a little too soon, good luck.
I do a lot of fungal biodiversity discovery and analysis, and just like having the right cereal dilution so that you can actually get those slow growers. But it actually comes back to a really important point, like reading your papers and knowing a little bit about your work now, having looked at it. A lot of us might take like a molecular approach to answering questions. Let's sequence all the ITS. Let's get a bunch of ITS data on fungal community assemblage in the soil. That just tells you what DNA is there. It doesn't tell you what the more active fungi are in that community, which ones are parasitizing which, because there was a number of mycoparasitic fungi from your studies. And I wonder, is the presence of this masking, or is the presence of this mycoparasite the reason we don't see a higher incidence of some of these other fungi, because these mycoparasites are eating whatever there? So putting this collection together is really, that's the complement to the DNA-based approach. You need a culture-based approach too to complement it and be able to actually carry out those experiments in a more formal lab setting.
Yeah, and this is something I love to highlight because I love bioinformatics and I could do it forever, but there are things that you just can't do.
Yeah.
Unless you have the microbe in your hand.
Yeah.
Which is why this process was so important for us to carry out. And I think the whole team is very proud of the collection we were able to put together. We ended up collecting a little over 600 isolates.
Oh, wow. That's a pretty good collection.
Yeah. It was a lot of work. I have pictures of our undergrad with just stacks and stacks of petri dishes.
Yeah.
It was wild. And from that collection, we were interested in looking at which ones had the highest likelihood of being parasitic to nematodes. And we decided on a few initial tests for screening. And it's interesting that you mentioned that a lot of these fungi are also mycoparasitic, because one of the tests that we did was whether or not the fungus could break down chitin, which every fungi has to break down chitin because they have chitin. So they need to be able to do some body recomposition, I call it.
Yeah.
But we were interested on whether they were able to produce it extracellularly and put it out into the environment, because same as fungi, nematodes also have chitin. They use chitin to protect their eggs. So basically the eggshell of a nematode has a thin layer of chitin. It makes it very resistant to the elements. And that's why you'll find that nematodes are hard to manage sometimes because their eggs are very resistant, so they can last in the soil for a very long time. So if you can find a fungus that can break down the eggs before the nematode even hatches, then great, you have an amazing biocontrol because the nematode is toast before it even has a chance to do any damage. So we tested for the production of chitinases. We also tested for the production of proteinase. And we tested for whether or not they could actually colonize nematode eggs. So we filtered out our collection. We narrowed it down from 600, right? We start at 600.
Yeah.
We lose a bunch of them, unfortunately, to storage.
Yeah.
So we tried two different storage solutions. Like we did your regular glycerol stock, and then we tried a sand method that you can keep them at room temperature within sand. And if your fungus sporulates well, then it lasts in that sand stock. If it doesn't, then bad luck. So we lose a bunch of them in storage, and that's helpful because even though it's sad that you don't have a good long-term storage, that you don't have to continue transferring into petri dishes.
Yeah, and you also don't have to test 600 strains.
Yeah. But also from a biocontrol development perspective, you also want something that is going to be amenable to long-term storage.
Oh, yeah, you want something that's durable. That makes a lot of sense.
So after that, we did all of these in vitro tests, and we tested first in the petri dishes for these extracellular enzymes and the things that passed that first filter. Then we tested with the nematode eggs because basically you need a lot of nematodes to be able to do these tests.
Yeah.
So the more we could narrow down our collection to something that was manageable, the better. And we found that a bunch of them actually did show good activity. And I believe it's at least 10 of them had enough activity that we were like, maybe it's time to talk with our tech transfer office to see if they think this is
Yeah.
worth.
Pursuing, yeah.
Yeah. And we were pleasantly surprised to find out that indeed it was of interest. So we now have 2 provisional patents for the strains. But what interesting thing from this whole tech transfer process was really learning about what industry is looking for in strains that they're interested in pursuing. Because as a scientist, I'm not thinking about all of these things. I just want to know, does fungus eat nematode? Yes, no, how, why?
Yeah.
And they're like, all of that is cool, but does it grow at 32 degrees? Because we're not touching that.
Yeah.
So one test you do to see if a fungus has human pathogen potential is you get them to grow at 37 degrees. And then if it does, it's probably not a good idea to continue to work with.
Yeah, we do some of that stuff too. And it's obviously there's going to be a lot more fungi that can grow at 37 that are not known to be opportunistic human pathogens. But if we're talking about Aspergillus or something that the genus is a known history of problems with humans, then you're like, okay, I'll stay away from that. But there's a number of trichodermas, for example, that will grow readily at 37. But you don't see a lot of reports of trichoderma infecting humans. And maybe there's some examples out there. I don't want to say that I exhaustively searched the literature. But I do want to kind of pivot a little bit to your time at Penn State in the Department of Plant Pathology and Environmental Microbiology, because we only have a limited amount of time. And you were just talking about these bioassays using actual cultures of fungi. A lot of your work at Penn State, from what I can see, you're doing comparative genomics of plant pathogenic bacteria. I won't judge you for working on prokaryotes, but I will. And then you're also looking at microbial predators and soil health. You want to talk a little bit about your work there at Penn State?
Yeah, so I started at Penn State right out of my PhD. And in the beginning, I started working with organic farmers and tomato production in organic farms. And the idea was to try to understand what micropredators are present in the soil of these farms and whether or not we could change the composition of the micropredators in the soil through organic amendments. So, micropredators, the way it's been defined for the project, it's basically microbes that will eat other microbes or that would kill them. Because included within the micropredators are like bacteriophages, which I wouldn't say they eat bacteria, but they kill them.
Or at least infect them.
Yeah. So I worked for a year in that project, and really a lot of the initial work was establishing a relationship with the growers, communicating what we wanted to do, enlisting them to collaborate with us in this project. So it was a lot of extension work and getting the project off the ground and less working with the data that came out of the project. Because after a year working in that project, I quickly move on to the lab I'm working currently. So for that previous work, I think what I learned most was really building relationships with a different population of farmers than what I had done in the past. Because although I'd worked with farmers in the past, I think row crops work in a very different way than, say, organic tomato farms. I was used to big acreage. Most of the farmers I worked with in row crops have a whole different career aside from being farmers. They're farmers and nurses. I've met farmers that are insurance people. I've met farmers that have are teachers. It was interesting moving into these smaller farms, way smaller farm that are organic, where the diversity of crops being grown there, it's very different. The management is very different. So I really learned a lot about a different cropping system that I was used to. And now I basically I say I miss the field work because I don't get a chance to do as much as I did throughout my PhD and my first postdoc position. But now I'm just a computer person.
Yeah. Those modern tools are so essential for being able to navigate industry or government jobs. You need to have the computational side of things. I saw one of the recent papers you were co-author on with Kevin Hockett and Carol Lee Bull, and this idea of translational microbiomes is really an interesting concept that we use these knowledge of the microbiomes to enhance plant health through microbe-aware crop management practices and just looking for stress, relatable community ecology patterns. I thought it was really interesting to take all this information and kind of put it into a framework outside of medicine. So I found that paper to be really interesting. Do you want to talk more about it or?
Yeah, so we purposefully chose to use the translational language
Yeah.
to draw from what is most commonly known as translational
That's
science,
it.
which comes from medicine.
Yeah.
And to a sense, we also do medicine, it's just different.
Plant medicine.
Exactly. So we wanted to keep the language because if we really want to apply the One Health framework, we need to be able to speak the same language.
Agreed. Yeah.
So that was a very purposeful decision from the group. And the idea of the paper really was trying to highlight the successes of the use of microbiomes for managing plant health. And I think during the process of writing this paper, we had people from very diverse backgrounds, and we quickly realized how difficult it was to agree upon the language of things.
Yeah. I saw there was a big table of definitions, which is actually really useful because you want to all be on the same page about that. But I couldn't help but think, just thinking about your publications you got out of your master's and your PhD work on corn and then on soybeans. This perfectly set you up to be a co-author on this paper because those were specific examples of how you're leveraging and monitoring the stress-relatable communities and community ecology patterns in corn systems or in soybeans and using that to inform management decisions. So it was perfect.
And I think there's so many ways in which microbiomes can be informative in contexts that are difficult for our classical approaches. So think about fastidious microbes. Sometimes there's just things that you can't culture or that are just difficult to culture or that are in such low abundance in a system that you're going to have a hard time identifying them until they become a problem. But if you think about the community as a whole and you can identify their more common partners. Let's say your pathogen really likes associating with this group of other microbes that are in higher abundance and
Yeah.
are readily identifiable. You may be able to catch these pathogens earlier just by understanding who they associate with.
Yeah. Yeah. No, that's great. And I think, as with a lot of things, you get these new terms and these new tools. I never heard the word plasmidomes until I saw that on your work. But it come back to this omics-based approach. People say, oh, I do genomics, or I do this. And to the general public, they're like, okay, that's just a word, but what does it mean and how does it help me? And I think, though, the idea of the translational framework is great because it's like saying, look, microbiome is a word that means this, but this is actually how we're using it to actually make manager decisions. Because if you don't follow up with how you're using it to make manager decisions, you've lost the general public on it.
And oh man, I really love the Ig Nobels
Yeah.
because there's so much research that it on its face, if you present it to the general public, they think that's absurd.
Yeah.
Why would you research that? How is that interesting to you? But I think everything that we research, it's either interesting or valuable in its own way.
Yeah. Oh, it has value. It's just that sometimes you have to wave your hands to communicate that. Other times it's obvious because it's intuitive.
But for example, for the plasmidomes, I've had people ask me, so plasmidomes basically is the study of the plasmids that are within an environment, and basically plasmids are ways in which microbes can move around genetic material and share genes.
So we're looking at whole bacterial communities and their collective plasmid collection.
Exactly. And people may say, okay, that's cool. You know what plasmids are there. Now what? And I think about if you know what plasmids are there and you know their structure and you know that they carry genes that have to do with resistance to antibiotics, and all of the plasmids that carry this resistance look the same, can you target them in some ways
Yeah.
and remove them from the environment so that you reduce maybe the speed at which resistance mounts and propagates within a community? So even if there's 100 steps in between what you're doing and the solution, we all have to start somewhere.
We're actually out of time, unfortunately. I've really enjoyed talking with you today, and I've gotten to learn some things and get to know you a little bit better. So I certainly appreciate your time, and thanks so much for joining us.
Thank you for having me.
You've just heard from Dr. Melanie Medina López, postdoctoral researcher in the Department of Plant Pathology and Environmental Microbiology at the Pennsylvania State University. As with all our episodes, be sure to check out the show notes immediately following the episode description for links to Dr. Medina López's research. I'm Matt Kasson, host of Plantopia. Thanks for listening.